Dear students, this is Dr. Pooja Thakkar, a biology expert. This is the 8th session of cell biology, that is on centrosome, cilia and flagella. This video is under the channel called Inside Biology. So do like it and subscribe the channel. Now, in the last session, we have discussed regarding microtubules as a specialized structure of cytoskeleton. Here we need to show the detail that how and exactly at where these kinds of microtubules are produced. Now we need to discuss regarding centrosome first. These are the organelles or a particular kind of area into the cell which contain two cylindrical structures called centrioles, which are cartwheel constitution, and they are located perpendicular to each other. Each and every cell is having one centrosome, that is two centrioles in them, and they are serving as the main site for microtubule production. So this particular center is also called as the microtubule organizing center, that is MTOC, specifically at the time of cell division process where the microtubule formation is needed. Okay, now each cell is bearing only one centrosome, but during the cell division time period, the centrosome multiply by binary fission, or else in the other language you can say a parent microtubule is forming a daughter microtubule, as you can see in this particular diagram. The first the yellow color portion is called centrosome, where actually the green colored tubular structure is present, which is centriole, which is basically serving as a parent or a mother centriole. During the course of cell division process, it will form the daughter centriole, which is shown over here. Okay, and so in other words, you can say the two centrioles are forming the other two centrioles. Okay, and these kinds of centrioles are travel to the opposite pole of the cell, and in other words, you will be able to notice that the centrosomes are now present at two locations, at two poles of the cell, or else you can say the centrioles are basically located in the perpendicular manner on both the sides of the cell, on both the poles of the cell, and they are orienting microtubules, as we have discussed in the last session. You can see again that video in which you will be able to notice the microtubule formation, the polar microtubule as well as the astral microtubule, which are attached to the chromosome for the exact segregation of genetic material between the two cells. Now here we need to discuss regarding the detailed structure of the centriole. Now how this particular centriole is made up of, as you can see in the diagram, it is a tubular structure which is made up of peripheral arrangement of the fibrils. So these are 9 peripheral triplets, as you can see in this diagram. Okay, the dark color structures called the trimer, or you can say the triplet. These kinds of 9 triplets are present at the periphery, which are very bold in this particular diagram, and they are typically called peripheral microtubules. The central proteinaceous part is called hub, as you can see in this diagram, right? Hub is labeled over here. So the centermost part is the proteinaceous material. Now the next labeled is spokes. What do you mean by that? The peripheral fibrils which are attached into the center at the hub by using the radial fibers or radial spokes. Okay, so the peripheral tubules are attached to the center by the structures called spokes. Okay, so this is the basic pattern of each kind of centrioles. And now what the next question come in our mind, where these centrioles are located. So these centrioles are playing a very major part of cilia, flagella and other kind of spindle fibers, or in other words you can say microtubules. So these are the structures which are basically present in the formation of cilia, flagella and spindle fibers. So now we need to discuss regarding cilia and flagella. Okay, so cilia is basically what? Cilia are hair-like projections, narrow shaped outgrowth on the free surfaces of the cell, and they are mainly aiding in the movement of substances from one place to another place. Okay, so they are typically present on the cells in the ductal system, and they help in traveling of material in any one specific direction. In other words, you will be able to notice the flagella. Flagella is basically comparatively, you can say, longer than cilia, and they are mainly involved in the motility work. Okay, so cilia are working as propellers for the movement of material as well as movement of cell or movement of surrounding fluid, whereas flagella is mainly involved in the locomotion. Now, flagella of prokaryotic cell as well as in the eukaryotic cell are structurally very different. Eukaryotic cells obviously are having comparatively complex structure of flagella. Now, what do you mean by the basic structure? As we have discussed earlier, that this particular structure is common. Now, which kind of pattern, the same pattern or a little bit different pattern, is present in cilia and flagella, that we need to focus on. Okay, so in the cilia and flagella also you will notice this kind of peripheral microtubules, but they are exceptionally forming the dimers at the periphery, as you can see here, in the form of triplet. There they are present in the form of dimer, and in the center also you will find central microtubules. Okay, so now we'll look at that. So these are the cross section. This is the cross section of cilia as well as flagella. Both are nearly having the same structure. As we have discussed, they are containing peripheral microtubules, which are doublets, as you will be able to notice. These doublets are attached to each other by using a bridge like structure, which is called interdoublet bridge. Okay, so these two labels are perfectly clear. The outermost boundary of the peripheral microtubule is labeled as plasma membrane, obviously, so that is the membrane of cilia and flagella. Okay, now the red colored structure which are basically present, which is segregating the core structure with the periphery, is called central sheath, which is present on both the side, on the left as well as on the right. Okay, now that kind of central sheath contain the central core part, which is always called hub, as we have discussed earlier, which is the proteinaceous material. So the center part is called, whatever red color is present, into which whatever center part is present, that is called hub, into which you will be able to notice the central microtubules. They are two in numbers. Okay, and they are singular, they are not at all doublets, they are singular. They are attached to each other, again by using or forming the bridge. Okay, but now what we need to discuss, the last label, that is radial spoke. As we have discussed earlier, radial spoke is mainly involved in connecting the peripheral tubules with the center. So here also it is doing the same task. It is attaching the doublets with the central sheath so that it can get attached with the center, that is the hub portion. Okay, so these are the labels and this is the common pattern. Okay, but now in this particular case you will be able to notice that this is the pattern which is called 9+2 pattern. The general pattern we have discussed earlier, in which the central microtubules are absent, that is called 9-0 pattern or 9+0 pattern. But typically in cilia and flagella, the pattern is called 9+2 patterns. Okay, so this is all about cilia and flagella. You can you can read each and every line in detail and ask the doubts in the comment box. Kindly do like the video and subscribe the channel, or you can also contact on the number, that is 990-980-4647. Thank you so much. And this is the last session of the chapter called the cell of standard 11. In the next sessions we'll discuss regarding the biomolecules which are present in the cell and how they are playing a crucial role for the sustainment of our life. Okay, thank you so much. Bye.